So when we're done replicating a cell, we are replicating a chromosome in a eukaryote such as ourselves, we run into a few problems. The major issue is that on the lagging strand, so let's say replication is overall in this direction, okay? Here's the last primer to be put down. So the primer is put down, and we replicate a new piece of DNA, okay? Then we come back through and we erase the primer. But because there's no more DNA down here to attach enzymes to, DNA polymerase cannot replace the primer with DNA. So what happens is this piece of the chromosome goes away. So the lagging strand gets shorter every time the cell divides. Now that's usually not a problem, because at the ends of our cells we have DNA that I think of as sacrificial DNA called telomeres. It's DNA that doesn't code for anything. There are no genes there. So we lose that DNA every time our cell divides. And it doesn't hurt us because that DNA doesn't do anything. So with every replication, that piece of the DNA gets shorter and shorter. That piece of the chromosome goes away. Now, the issue is, let's say you have a chromosome and it's dividing over the course of your lifetime, and here's a telomere on the end of it. That's junk DNA. Well, every time your cell divides, it gets shorter and shorter, and eventually you use it up and you start actually removing actual genetic material. And that is one of the sources, we think, of aging. Why, as we get older, we're slower to heal. Why different symptoms of aging occur might be because we're losing genetic material as our cells have divided and they lose genetic information. Now, germ cells, like stem cells that give rise to new sperm all the time, or cells that give rise to your skin all the time, they have an enzyme in them called telomerase that may help them to rebuild the ends of those telomeres, so they can rebuild telomeres. Now, unfortunately, cancer also can have this enzyme. Telomerase, erase, that ACE ending lets you know it's an enzyme, and in this case, it's an enzyme that rebuilds telomeres. So as those telomeres get shorter, they build longer ones. And so you might think, ooh, that would be a great thing to have. You could just like take a telomerase pill and prevent yourself from getting old. But unfortunately, in experiments where we inject mice with telomerase, it just causes cancer. But cancer cells can divide and divide and divide over and over and over again and take over a body, basically, and they don't get old. Whereas a regular human cell can only do that about 10 times. And then the lineage ends because the genetic material is lost. But cancer cells don't have that problem. So there's some chromosomes there with the telomeres labeled. You can see them glowing. There's an interesting case, and I invite you to watch this video, on Henrietta Lacks. Henrietta Lacks was an African-American woman who got cervical cancer in the 1950s. And went to Johns Hopkins University for treatment. But back then, scientists would often take tissue samples without even telling you they had done it. And at the time, they were trying to grow human tissues in the lab, and they were having a hard time. But her cancer cells actually were able to be grown in a lab. She died, unfortunately, right after that from cancer. But her cells still live on today. And if you ever see a paper, a scientific paper with HeLa cells, that comes from Henrietta Lacks. And there's also a lot of ethical implications with her story, because a lot of companies have made lots of money off of her cells, but her family didn't even know it was happening, and they were also very poor. And there's a really good book out there about it called The Immortal Life of Henrietta Lacks, and I think Oprah made a movie about it as well, and I really recommend you check that out. But her cells grew in a lab, and they had telomerase that allowed them to divide and divide and divide. And if you watch this clip, and I'm going to put it, I'm going to have questions on this clip in the on the test, so please watch it. Pay attention to how many Empire State Building's worth of her cells have been produced. It's amazing. So we've talked about chromosomes in this chapter. We've talked about DNA, I should say, in this chapter. I want to relate it back to the idea of chromosomes then. So DNA is a double helix, as you know, and it's wrapped around proteins. So here, this thing right there, that's a protein. They're called histones. And that DNA wraps around histones, kind of like thread around a spool. And that's how it's able to fit inside of a cell. Because you have like 6 feet of DNA, but you wrap it up really tight around these proteins and it condenses down. So if you wrap up DNA a whole bunch and wrap that up even more, you get a chromosome. Okay, so as we see here. Now, typically in your cells, the DNA is a little bit unwound. And so it would look more like this, kind of like cotton candy. But during mitosis or meiosis, the DNA curls up really tight and forms the chromosomes that you're most familiar with. And so when we talk about chromosomes, when we talk about mitosis and meiosis, we're talking about a double helix of DNA that's coiled up really tight. And we'll talk more about that in the future. All right, so in the next lecture, you're going to hear about genetic engineering, and we're going to go on into how proteins are made using genetic information, a topic known as gene expression. So that's where we're going next.